Hello everyone. So I decided to record this lecture on bioinorganic chemistry to get a chance to go through it this semester. But this is a topic that I'm very personally interested in, and I could easily teach a full course on this. So this is just a very brief overview of this topic. So I'm going to skip over all of this class stuff because it's not relevant to us anymore. But just to start off, what is bioinorganic chemistry? So as the name implies, this study involves the investigation of metals or inorganic elements in the context of biology. So bioinorganic chemistry is the study of naturally occurring metals, as well as metals that we introduce into the biological system in the form of probe molecules and drug molecules. So we'll see a couple examples of actually all of these cases in today's lecture. So you probably know that metals are essential in living systems and that they carry out many different functions. So some of these metals you're quite familiar with: sodium, potassium, calcium, iron, copper, zinc. But there are others that you might not be as familiar with. So things like chromium, molybdenum, vanadium, tungsten, they all play very specific roles in many different biological reactions. So that's why they're important. That's why we study them. If you've taken inorganic chemistry, and I think most of you guys have, you know that metals can differ in many different physical and chemical properties. For example, they have different coordination geometries. They have different oxidation states. Boost acidity. Kinetic lability of bound ligands. Which basically just means how easily the ligand comes off and on to the metal. So the exchange rate and other characteristics. Okay, so compared to organic elements like carbon, because metals have such different properties, they can be taken advantage in biology for catalyzing biological reactions. So here's a nice table showing what are some of the main elements that we study in bioinorganic chemistry. So the metals that are highlighted in for this red color are metals that are naturally occurring and that are very abundant. So these are the alkaline metals, so you can see them here. And the ones that are highlighted in green are used as either probes or drugs. So a really popular one is platinum that I'll talk about sometime at the end of this lecture. So this lecture is organized in terms of functions. So we'll go through different functions of metal-containing proteins and then giving some examples to kind of illustrate how they work. So one very important function of metalloproteins is involved in O2 transport. And there are three main classes that have very different active site structures. This one you probably know about, hemoglobin and myoglobin. So these enzymes, or proteins actually, they're not enzymes, they use a heme cofactor and they're found in vertebrates. So lots of animals use this heme for respiration. And here is the active site structure of these proteins. So they contain these iron-containing sort of porphyrin type units and there's an axial coordination of a histidine side chain. So in the unbound form, notice that the iron is the +2 oxidation state. And so in this form, the iron is a little bit too big to fit within the planar structure of the porphyrin. So we see how it's kind of puckered and is sticking sort of below the plane of the porphyrin. However, when it binds O2, the O2 actually gets reduced to superoxide. So that's O2 minus. And actually, yeah, that's correct. And notice that now the oxidation state has changed. So iron 2 has given an electron to oxygen, so it's now iron 3. That makes the metal smaller, and so it can move toward the plane of the porphyrin. So you see in this cartoon here is a very flat molecule. And there's an important consequence of that, because notice that the axial position has a histidine coordinated, right? So when the histidine moves towards the iron center, it's going to pull on the bonds that are connected to it, which leads to a conformational change in the protein. And this is what allows it to achieve cooperative binding. I didn't show the other subunits, but in hemoglobin, you have actually 4 of these units. So once one oxygen binds to one of the iron, it activates the other irons to bind oxygen with even higher affinity. So this is a really neat system where it shows this cooperative binding activation. So mammals use hemoglobin and myoglobin. Hemocyanin has a very different active site structure. It has a same function, right? It's used for transport dioxygen. But in this active site, it uses a dicopper center. And it is found in mollusks and arthropods. So things with exoskeletons. So this active site, it's very different from hemoglobin. The two copper in the reduced forms in the copper 1 state, and you have lots of imidazole side chains that's supporting the metal center. When O2 binds, it actually reduces the oxygen to the peroxo, which is 2 electron reduced. So now it's O2 minus, and the electrons are coming from copper. So now copper is oxidized to copper 2. And notice this equilibrium arrow, because it can bind O2, but more equally important is that it can release O2. That's how it serves as an O2 transporter. And the last example, which we actually talked about earlier in the semester, is hemoerythrin. This one uses a diiron center. And it's found in invertebrates, such as worms, marine worms. So structurally, it looks a little bit like the hemocyanin active site, except that you have irons. And in addition, we have bridging carboxylate groups. So these ligands are really holding the metals in place. You want a really rigid structure. You have a bridging hydroxy group. And there's also a change in oxidation state upon binding, right? So iron 2 has reduced the O2. Actually, this also provides another electron to the hydroperoxo. In this case, hydroperoxo. So that's HO2 minus. The hydrogen bonding interactions helps to stabilize the structure so that it can be a reversible transporter. So what's important about all three of these O2 transporters is that they have to achieve this delicate balance where they can bind O2 to iron or copper without undergoing irreversible electron transfer leading to OO bond cleavage. So if this bond gets broken, then it's going to react with something else and you lose the O2 molecule, right? So the trick that nature has figured out is how to stabilize the O2 molecule and allow it to be released so that this could be a reversible process. There are other proteins that do that don't just bind, they will bind to O2, but they'll activate it and they'll use that O2 molecule to carry out other reactions, which we'll talk about soon. So those are examples of O2 transporters. There are metalloproteins that are only involved in electron transfer, so they only move electrons around. These current carriers usually pass their electrons to or from enzymes that require redox chemistry in order to perform a specific function. There are three major classes of these electron transfer enzymes. This one we've seen before, the iron sulfur clusters. They are found in, for example, succinate dehydrogenase, or aconitase. And these protein names probably sound familiar because they come from the TCA cycle. So this is a very important class of enzymes for a number of reasons. And there's a variety of different structures. You can have one that has a single iron site, so these are the monoiron, and the nomenclature is just based on how many sulfurs and how many irons. So this one would be like a 1 iron, 4 sulfur cluster. This would be a 2 iron, 2, 4, 6 sulfur, and so on. The second class are cytochromes. They can be further classified as type A, B, and C, and they differ in the heme group. So they look kind of like the structure of the heme in hemoglobin and myoglobin, except that some of these ligands around the porphyrin ring are a little bit different. But you can see in this X-ray structure of a protein, you've got your heme and you have different axial ligands, and the different types are just based on exactly the identities of these peripheral groups around the heme unit. Metals are sometimes used to help stabilize protein structure, so they play a structural role. Metal binding can help stabilize protein folding. And conformations, or maintain conformations. The most common metals that's used for structural functions is usually zinc 2+, and they're called zinc fingers. Zinc fingers were originally coined to describe the finger-like appearance of transcription factor 3A. That's when they were first observed. Which appears to be directly involved in intercalation with DNA. So let's say which intercalate It's a really nice cartoon. Actually, I don't know if this is a rendering or an X-ray structure. But this is supposed to show the DNA double helix. So you see that nice helical coil. And then the blue is the zinc finger. And the green spheres are the zinc ions. So these zinc ions are allowing this protein to assemble in a way that looks like little fingers. And the fingers are wrapping themselves around the DNA coil. So that's what we call the intercalation interaction. So if metals and proteins serve a catalytic function, we call those proteins metalloenzymes to indicate that they catalyze reaction processes. So metalloenzymes are a subclass of metalloproteins that perform specific catalytic functions. This is a table to kind of summarize a few classes of enzymes. So we can classify them based on what they do. Some do hydrolysis chemistry, some are involved in nucleotide transformation, isomerization, photosynthesis, nitrogen fixation. These are some examples, specific examples, and some of the reactions that they catalyze. So we're going to go through a few different examples, give you an idea of the flavor of enzymes that are possible. So in the hydrolytic enzyme category, these enzymes catalyze the addition or removal of water So some notable examples include carbonic anhydrase, which promotes the hydration of CO2. And that reaction is pretty simple. It's just CO2 plus water, and it makes carbonic acid, H2CO3. There are enzymes called peptidases and esterases, and as the name seems to suggest, it cleaves ester bonds and amide bonds. So these are hydrolytic enzymes, so that means they can take things like, or substrates that have an ester group, utilize the water to do a hydrolysis, which will lead you with the carboxylic acid and the alcohol. The phosphatases do similar chemistry. They hydrolyze phosphate esters. So phosphate esters have an OR group in addition to oxygen atoms attached. So hydrolysis of this molecule will leave alcohol plus phosphate. So many of these hydrolytic enzymes contain the zinc ion, zinc 2+, because coordination to the metal lowers the pKa of the metal containing ligand. So for example, usually the pKa of an alcohol is like 16, but if you're able to bind the alcohol to a metal, this pKa is going to be lowered, right? Because it's stabilized, that resulting conjugate base, the O- will be stabilized by interaction with the metal, with the M+. The exact pKa is, of course, is going to depend on what metal, what oxidation state, but in general, the pKa value will go down, which makes it easier to deprotonate. And zinc is such a special ion because it avoids undesired electron transfer chemistry, because zinc 2 plus, you know your electron count, is a D10 metal ion. So it does not have any accessible redox states. Okay, so if you have a closed shell configuration ion, that means it doesn't want to gain an electron or lose electron. So that means you usually see zinc as only a 2 plus and not zinc plus 1 or zinc plus 3, because that would not be a complete a closed shell configuration. However, this is not always true. There are some hydrolytic enzymes that utilize other metals that can be redox active. Manganese, nickel, and copper, they can cycle between these plus 2 oxidation state, or they can be reduced or oxidized. Magnesium 2 plus usually stays as magnesium 2 plus, also a closed shell ion, but there are many different examples of hydrolytic enzymes using the Lewis acidic properties of these metal ions. Okay, so the first type of reactions are pretty simple, just adding water or removing water. You could have reactions that involve 2 electron processes, so we're going to increase in complexity. They catalyze 2 electron reactions that often involve atom or group transfer between substrates, and here are some examples. Cytochrome P450 is a class of enzymes that we classify based on their absorption at 450 nanometers. That's due to the heme group that they have. But these enzymes often do alkane oxidation, so they can convert some kind of alkyl chain to the corresponding alcohol, and they use that iron heme cofactor with O2 as a source of oxidant. So one of the monooxygenases will take one oxygen from O2 and transferring it to the substrate, and then the other oxygen will just be water. Another example is tyrosinase, and this enzyme controls the production of melanin. Melanin is a natural pigment found on our skin and in our hair, and it comes from polymerization of catechol. So this enzyme only is involved in forming the benzoquinone that goes on to make melanin, but it starts with copper 1, it binds O2, but instead of releasing O2 like the hemocyanin, it actually further coordinates to the tyrosine molecule, because you've got a nucleophilic group here that can bind to copper. And then in this very critical step here, the O2 molecule is broken and one of the oxygen atoms are transferred to this benzene molecule, so you end up with what we call the catechol molecule. That's a benzene with 2 OH or O minus groups. And then the last step involves oxidation of this catechol to provide the copper 1 reduced form of the enzyme, and it generates this benzoquinone, which eventually is going to become melanin pigment. Another 2 electron reaction is catalyzed by sulfite oxidase. This is present in the mitochondria. And it transfers electron to the electron transport chain. I guess I should probably squeeze in the reaction right here. So what this does is it takes the molecule sulfite, has this Lewis structure, and it converts it, it oxidizes the sulfite to sulfate to balance. We have two protons and 2 electrons. But what's interesting and that we haven't seen yet is that this uses a molybdenum cofactor. So this molecule has this kind of really interesting looking ligand with 2 sulfur groups and a couple oxygen atoms. But this is the cofactor that's doing the oxidation. Basically, one of these oxygen atoms are being added over. There are enzymes that remove oxygen from the substrates, and a really important one for living systems, and this is present in all living systems, is ribonucleotide reductase. It is responsible for the first step in DNA biosynthesis. Because all living things have DNA, this R&R enzyme is very essential. And the reaction it catalyzes, it's a very important one. So you probably remember the structure of the ribonucleotide. You got your sugar ring and your base and your phosphate group. And using a diiron cofactor, and O2, it is actually able to remove the OH group at the second carbon, okay, which is right here. So this is called the ribose. This is now the deoxyribose, which is the building block of all DNA polymers. Nitrite reductase catalyzes a key step in nitrate assimilation in green plants. So nitrate's Lewis structure is like this. It's got 3 oxygen atoms. It uses another molybdenum cofactor, except now it has 4 sulfur ligands. And it removes one of the oxygen atoms so that we convert this nitrate to nitrite. So some of the most complicated enzymes, metalloenzymes, are the ones that do multi-electron redox chemistry. So these reactions involve 2 or 3 electron pairs. And in general, these transformations sorry, I gotta squeeze this in here, have not been duplicated in non-enzymatic chemistry. Yeah, so synthetic chemists have gotten pretty good. We're able to, in some cases, mimic the biological reactions using a synthetic system. But because of how complex these multi-electron enzymes are and the complexity of the reactions, we've not been able to, in many of these cases, reproduce the same reaction under similar conditions. So here are the 2-electron processes that we've talked about: O2 reduction, catapulsidation. 3-electron processes, electron pair reactions, so a total of 6 electrons, includes nitrogen reduction, sulfate, or formation of H2S, and N2O reduction to ammonia. One example is cytochrome oxidase. It is involved in the last step of the electron transfer process. And the reaction, the overall reaction, is O2 plus 4 protons and 4 electrons. And what's really unique about this enzyme is that it contains a mixed metal system. It's got copper and iron, and it reduces the O2 to 2 molecules of water. So here is the X-ray structure of the active site. You can see that the 2 metal centers work cooperatively. So you've got your iron and your heme type group, or porphyrin group, and then the copper has different ligands. These are imidazole side chains, and the O2 molecule binds perfectly. That allows it to bridge between these 2 metals, and then through a complicated process, the protons and electrons are added to the O2, basically to split it into water. There is a lot of interest in studying how photosynthesis work and to basically reproduce it in a synthetic system. So a lot of people are studying specifically this complex, the photosynthetic oxygen evolving complex. And what this complex, or this enzyme, does is it takes water using a very special 4-manganese calcium cluster, is able to produce oxygen, but what's important is that it generates electrons. And these electrons can be used further down for other further downstream for other processes. So this critical step is catalyzed by this cluster as shown down here. You've got sort of this cube-like structure with the calcium in the light blue here. Here's an actually easier depiction of that. So you have this cube that includes manganese and oxygen ligands and the calcium, and also another manganese that's actually outside the cube. And that entire structure is supported by carboxylate residues in the protein. So super complicated. People are studying this system, but there are still a lot of questions about how it works, and trying to mimic that has been challenging. Another very intriguing enzyme is the nitrogenase enzyme. It is found in certain bacteria, and in certain bacteria they're able to reduce dinitrogen in air to ammonia. This is also called nitrogen fixation. So if you read the literature, you might hear that term sometimes used. And this is such a challenging reaction because normally if you take N2 and try to directly reduce it to ammonia, the activation barrier for that reaction is extremely high. But nature, again, has been able to figure out how to get around this using a multi-step process. So exactly how that works is still very debatable. The active site of this enzyme is believed to contain this structure. So you've got molybdenum, you've got irons, you've got sulfurs. It took a long time for scientists to figure out what this middle atom is, but the current hypothesis is that it's a carbon, and this was established through a lot of really advanced spectroscopy work. And because it's so complex, the actual mechanism of how that works is still a mystery. But there have been two proposed pathways, either the alternating pathway up here or the distal pathway. So I'll just walk through this very quickly. The alternating pathway just involves kind of alternating steps where you transfer electrons and protons to one nitrogen and then the other. So if you follow this, the N2 molecule binds the iron, right? So you've got this. And then you can protonate this nitrogen and transfer an electron to get to this structure. Then you protonate the second nitrogen, you protonate the first one again, and then you protonate the second one. And finally, you can cleave this nitrogen-nitrogen bond to make ammonia here, which leaves you with one nitrogen left. And then the last step is to protonate again, and then release your ammonia. So this is called the alternating pathway because you are kind of flipping between nitrogen number 1 and nitrogen number 2. Okay. And in the alternative pathway, it's been proposed that maybe it just happens at this first nitrogen first to remove it as ammonia, and then you can protonate the second nitrogen. So if we walk through this scheme, the first step is the same, you still protonate that first nitrogen, but the second step, you protonate the same nitrogen one more time, and then now instead of NH2, you can protonate it to get NH3. That's going to leave you with the iron nitride, which could then be further protonated once, twice, and then the third time. So, you know, lots of different biochemists and synthetic chemists are studying this reaction at the moment, but those are the two prevailing mechanisms. Okay, some enzymes catalyze rearrangement reactions, so they're just changing the placement of the chemical bonds. They do not involve any net change in the oxidation state of the substrate. So enzymes that catalyze 1,2 carbon shift requires a cofactor called cobalamin, or also known as vitamin B12, as a cofactor. This is such a really remarkable cofactor because it's the best studied organometallic unit in biology. If you're interested in organometallic chemistry, we have a graduate course here taught by Dr. Olaf Stargulis, but that's offered in the fall. But organometallic species are compounds that contain a metal-carbon bond. And if you look at this very complicated looking structure, the metal-carbon bond is right here. So instead of the iron that you are used to seeing, there's a cobalt in vitamin B12. And that cobalt is connected to a alkyl group. Reactions usually take place by free radical pathways initiated or catalyzed by homolytic cleavage of the cobalt-oxygen bond. Okay, so what's proposed is that this bond right here homolytically cleaves, so then one electron goes to cobalt, one electron goes to the alkyl group, so you have an R dot, and the R dot is what goes on and does the chemistry. It is an important, another important rearrangement reaction is catalyzed by conatase in the TCA cycle. In case you forgot what that reaction involves, is you take citrate, which is a symmetric structure, right? You've got two carboxylates, you've got the center carbon that has a third carboxylate with an OH. And aconitase uses the iron 4 sulfur cluster, and it moves that OH group to this carbon. Right, so it moved it from the center carbon to the second carbon, the alpha carbon. And this is a rearrangement reaction because we're just moving bonds. Right, the hydrogen here has now moved to over here, and then the oxygen has taken this place. Okay, so why do enzymes that are involved in multi-electron processes require polynuclear metal clusters? So you've seen examples where you could just use one metal to catalyze reactions. So the question is, well, why has nature evolved to use this very complex multinuclear, polynuclear species? So let's look at what's these answer choices and pick the best one here. A, there are multiple sites for reaction to take place. That can be true, but often you want selectivity, so I would say that that's probably not the best answer. The large clusters make the protein active sites larger for better substrate access. That has to do more, I think, with the active site environment. So if the active site pocket is very open, you can fit large substrate into it. It's very narrow, then you can't fit large substrates. So I don't think that necessarily having multi-metals is important. A single metal center would not be able to accommodate exchange of electrons of many electrons. So I'd say that's probably a true statement, because with a single metal, you're restricted to the number of electrons on that metal, depending on what it is, right? So if it's iron, usually you can access iron 2, iron 3, iron 4. So maybe you can have up to 2 electrons, or possibly even more than that, but especially for these 6 electron reactions, right? A single metal is not going to be able to accomplish that. So I say that's a pretty good answer. Multi-metallic cofactors are more difficult to degrade. I don't know if that's true. I think a lot of monometallic cofactors are pretty stable in the biological environment. Some enzymes can be replaced with single metal cofactors, so polynuclear clusters are not absolutely necessary. To my knowledge, I don't think that this has ever been shown, where you can replace the cofactor from a polynuclear cluster with a single metal complex. So I'd say I'll probably put X through that one. So the best choice here is that multinuclear clusters are necessary because you can extract more than just a couple electrons from that cofactor. Some metalloproteins or I guess not necessarily proteins, but metals, can be used for communications in biology. A really neat example is these magnetotactic bacteria. It uses magnetite, which is Fe3O4, as an internal compass by which to navigate. So this is pretty interesting to me. These organisms swim north, but also downward along the Earth's magnetic field lines to avoid high O2 concentration of the surface water. And because O2 is actually toxic to these bacteria, they want to be able to avoid being exposed to O2. And I think this is a image of that bacteria, and you could see that in the internal space of the bacteria, you see these orange spheres, which I think is supposed to represent the magnetite. Magnetite is insoluble. But because iron is paramagnetic, that means that it can actually sense the magnetic field of the Earth, and that actually helps it to navigate in the ocean. So pretty cool, it's got an internal compass. Earth metal ions such as potassium, sodium, potassium, calcium are used in biology to trigger cellular processes. So for example, rapid influx of sodium across the cell membrane is observed in the firing of neurons. calcium is an important secondary messenger. So that's why it's really important that we have a good balance of these metals, because if it's either deficient or if it's in excess, then you're going to be interfering with kind of these cell signaling processes. Some metals interact with nucleic acids, and we've already seen this in previous lecture. Non-specific interactions of potassium or of sodium and magnesium with nucleic acids shield the charged phosphates from one another. So you know that DNA, because it has phosphate groups on the nucleotides, it's actually a polyanion. And so by having a positively charged cation, you can actually have sort of this electrostatic interaction, and that helps to neutralize the anionic charges. So these ions actually stabilize this DNA structure. Telomere structures are characterized by non-Watson-Crick base pairing interactions and clustering of 8 guanine bases around the central metal ion. So you think of the metal here as kind of a template. So here's a nice depiction. We've got this potassium ion, and you've got this G quadruplex, and because of the Lewis acidic nature of this cation, the guanine nitrogens combine to the cation, and there are 8 of them that's interacting. So I think these 4 up here, and then the 4 on the bottom, so it fit perfectly to kind of template and to stabilize this G quadruplex. Okay, so because metals are so essential to biological system, it has to figure out ways to both store the metals as well as to transport the metals. So this is a very active area of research as well. For example, iron in bacteria, iron enters bacterial cells following chelation below molecular weight compounds called siderophores that are excreted by the organism. And in the next page, we'll be able to see some structures of siderophores. So chemically, they're very interesting molecules. They usually contain some kind of macrocycle, and then they have these side arms. And what's important about these side arms is that it's got these oxygen donors, and they serve as really great ligands. So what can happen is that when the siderophore is secreted in the environment, it's looking for iron specifically, and these oxygens can then bind to the iron atom, so it makes an octahedral complex, and then it solubilizes the iron so it can be absorbed by the bacteria. So they have very diverse structures. You see here's one siderophore, this one has a larger macrocycle ring. In this case, the binding unit is the carbonyl and the hydroxy group on this hydroxyamide. And here's a different one that uses a catechol. So they are chemically very interesting species. So bacteria must acquire iron from the immediate environment. And this is challenging because naturally occurring iron sources are insoluble. Right? So if it's insoluble, that means the bacteria can't just take it up. And so what it has to do is to chemically sequester the minerals from the atmosphere into a soluble form. Because once this complex is soluble in water, and so then it can diffuse in the water toward the bacteria. So how do we store iron? in mammals. In mammals, iron is bound and transported by the serum protein transferrin and stored in ferritin. And this ferritin protein can bind up to 4,500 iron 3 ions. And this is important because you need to be able to store in a compact form, right? You don't want to take a lot of space, but so you want to have efficient storing. And so here's an image of this, what this transferrin protein looks like. In the interior of the protein, the red that you see that's sort of peeking out, is a cluster of these 4,500 iron 3 ions. And what's cool about this is that in this form, this cluster of iron is actually very insoluble. However, because this protein kind of surrounds it and wraps it up, this helps to keep it in your blood, and so that solubilizes this normally insoluble iron storage. Copper is also important, and copper is a whole different system that controls its trafficking in the body. So copper is transported by the serum protein ceruloplasmin. Copper can be acquired from the liver through this protein ceruloplasmin, and then it's taken to the plasma, which then is used for, in this particular example, vasodilation. Okay, now switching gears. So all of those examples that we've talked about so far are naturally occurring metals in biology. However, because if you look at the periodic table, there are many other elements that are not utilized by biology, but as synthetic chemists, we can introduce new functions by creating molecules that utilize these metals. So here are some successful examples of metals that are used in medicine. The first one is called cisplatinum. It's a very simple molecule. It's got a platinum atom that's bound to 2 ammonia molecules and 2 chlorine atoms. And this is called cis because these groups are cis to one another. You can actually have the trans isomer, right, where the chlorine is over here and then nitrogen is over here, so they're trans to each other. But cisplatinum, in this stereoisomer, is the one that's biologically active. And what's important about this molecule is that it's a very commonly prescribed anti-cancer drug. The most incredible thing about this drug is that it cures testicular cancer in about 90% of cases when diagnosed early. I think this is still true that cisplatin is one of the most commonly prescribed drugs for cancer. So whether or not you're aware of this, this is still one of the best drugs in the market. And what's cool is that it's a cure rate. So if you're lucky enough to have early detection, there's a very good chance that you can actually be cured of testicular cancer. There are other cancers that this drug is used for. I don't remember which ones, but I think the most successful is for testicular. The problem with cisplatin is that it's actually also highly toxic. So patients who take this drug often have a lot of different side effects. Some of them can be quite severe. So there are other derivatives of platinum that are, for example, like oxaloplatinum, for example, is a similar drug but has less side effects. Other people are developing newer versions that might be better. Oranafin is another drug. This is used as an oral rheumatoid arthritis drug. And the key element in this molecule is the gold. So you've got this sugar that's been acetylated. There's a sulfur group that binds to the gold, and also a triethyl phosphine. And gold, if you remember from your inorganic chemistry, is a very soft metal, so it prefers to bind ligands that are also soft, like phosphorus and sulfur. It's a very polarizable nucleophile, so that's why it's used in this particular case. The last example here uses technetium, and this molecule is called cardiolite. It's a heart imaging agent. And the reason that it's used as a diagnostic agent is that it's selectively taken up by the heart. Because if you have a diagnostic agent that just goes everywhere in the bodies, it's not very useful, because then you see too much background signal. So for technetium, this cardiolite molecule, it's able to selectively localize in the heart. So it's got a very unique structure. The technetium's in the center. It's got these isocyanate ligands with these methoxy groups, so pretty simple structure. This is a radioactive drug, so the technetium is actually, I don't remember the isotope, but it decays and it produces gamma rays, and the gamma ray is what's being imaged when you take this, use it for heart imaging. Okay, so those were the examples of drugs that are used as medicine, or metals that have been used as medicine. And this last section is about what kind of tools and methods that we use to study bioinorganic chemistry. So what are some common methods? Spectroscopy is a very important one. Spectroscopic methods take advantage of the unique characteristic of inorganic complexes. Use of different wavelengths of light allow different transitions to be studied. So this is the electromagnetic spectrum. So on the far left here, you have very high energy radiation, the gamma rays, X-rays, and then UV. This is the visible range, and then you've got the IR. These are now the long wavelengths. IR, microwave, radio waves, long waves. And this table here shows you what type of electrons these energies will excite. So if you're looking at nuclear transition, you can use gamma rays or X-rays. You're looking at valence electrons, UV vis, or IR, it's for vibrations. And then if you're talking about NMR, that's the radio waves, nuclear spins. So UV vis spectroscopy is useful because many inorganic complexes exhibit D to D and metal ligand transitions that occur in the UV visible region of the electromagnetic spectrum. Here are some of the common chromophores that we've talked about. So the heme unit, iron with nitrogen ligands, iron sulfur clusters, copper and nickel probes, the blue copper protein. These are the different wavelengths that they appear, so usually in the visible to UV region. And then these are the different transitions. So some of them come from the pi to pi star. So for the heme, you've got these very flat aromatic molecules. So you can have the homos, that's pi. The pi star is where the excited the antibonding orbitals. Some of the transitions are due to excitation within the D manifold, so we call those D to D. Some of them involve transitions of the ligand to the metal, so that's LMCT. I guess there are no MLCT examples in this chart here. Another useful tool is electron paramagnetic resonance spectroscopy, also called EPR. And this method works for studying systems that have unpaired electrons. So for NMR, because of the selection rules, you usually only use it to study diamagnetic compounds, where the spin total is 0. So EPR is the opposite. it's used to study systems that have non-zero spins. It measures the absorption of microwave radiation by a paramagnetic system, and many first-world transition metals have unpaired electronic configurations. So here's the typical output of an EPR experiment. So you have your sample, which we put into a tube that looks like an NMR tube, but it's a little bit thinner usually. And it looks at absorption of microwave radiation. And so you'll get something like this, right? If it absorbs, there's a transition, you see that transition there. But normally you would take the first derivative so that you can see the field at which the transition happens. So that's usually what you will see as the EPR spectrum, this first derivative form. So yeah, spectra are typically shown as derivatives of absorption spectra, can provide information about metal oxidation state, spin state, and electronic environment. Interpreting EPR spectra is much harder to do than interpreting NMR spectra. So often you need to have a specialist to interpret very complicated EPR data. You can use high energy radiation, X-ray absorption spectroscopy. So this has to be done usually at a synchrotron because we can't generate that intensity of light using just normal laboratory equipment. It is based on the absorption of X-rays by core electrons to higher energy levels. The core electrons are the 1s level. It can determine oxidation state and coordination environment. So this is what you'll typically get from a XAS type experiment. And this plot is shown here as the energy of absorption, well, energy on the x-axis, absorption on the y-axis. And we usually divide the spectrum into two parts, the xanes and the xavs. So the xane stands for X-ray absorption near edge structure. And this shows you the energy of absorption from the s orbital. And so this gives us information about the oxidation state because the energy at which that happens will be different, for example, between iron 2 versus iron 3. But just by looking for the xane peaks, you can sometimes assign the oxidation state. In this region here, you call the x-as, which is X-ray absorption fine structure. You see a bunch of these peaks. But we can actually fit this data to figure out what atoms are connected to the metal center. If we're studying, let's say, the porphyrin unit, you might see if you fit this data to neighboring nitrogens, because it's coordinated by nitrogens, and then using that data we can actually figure out the distance between the iron and the nitrogen. So the x-axis region is actually super important because it gives you so much information about what's surrounding that metal center. But these type of experiments are not easy to do because you have to go to a synchrotron and you have to have a lot of expertise in both measuring the samples and interpreting the data. This is why XAES is not done on a routine basis, but it is a very, very powerful tool. If you do any type of iron chemistry, whether in the synthetic chemistry or particularly in biological chemistry, there's a very important technique called iron-57 Mossbauer spectroscopy. It is based on the absorption of gamma radiation by the 57 isotope iron nucleus, and it probes iron atoms specifically, no signals from other metals. And it's so useful because let's say you're studying a protein that has a lot of different metals in it. So you have iron in addition to iron, you also have, let's say, manganese or cobalt, nickel, other metals that can absorb in using other experiments. But because of the energy of excitation of this iron-57 isotope, you're selecting for only the iron atoms, and you can see the environment of the iron. Here's what you'll typically see in a Mossbauer experiment. So in the absence of quadruple splitting, you'll just see one peak, and then we call this the isomer shift. If you have quadruple splitting, sometimes you can see 2 peaks, or in the presence of a magnet, you can see more than that. So interpreting these spectra can be kind of complicated. But what's important is that it provides info on oxidation state. As inorganic chemists, we are always talking about oxidation states. The ligand environment, whether it has a symmetric or an asymmetric environment, and can differentiate between chemically distinct iron centers. And then this very last part, which I will finish with, is studies using synthetic protein models. So in the previous part of this lecture, you've seen many examples. We have very complicated structures. The proteins are really large, usually thousands and thousands of atoms large. And so it's very hard as chemists to study them. So one approach that's been used is to just make model complexes of just the cofactor, so that we can probe the structure and study the chemistry. So due to the complexity of metalloproteins, they can be difficult to study using traditional biophysical methods. The active sites of metalloproteins can be mimicked in a small molecule model. However, achieving both structural and functional mimics can be challenging. So I just wanted to end with kind of what I did during my PhD that's relevant to this topic. Basically, the protein that we were studying, or the enzyme actually, is called soluble methane monooxygenase. It's a really cool enzyme. It's found in bacteria. This was isolated from the bacteria that's found in the hot springs of Bath in England. But what's cool about this enzyme is that it does a very simple reaction, and it takes methane to methanol using O2, electron sources from NADH and hydrogen. And the reason that we care about this reaction is because we can't do this in the lab under mild reaction conditions. So there are ways that we could chemically convert methane to methanol, but usually it's very high energy, it's not selective, so it's just not a very good chemical process. But however, nature's been able to figure out how to activate methane in a very selective manner under conditions that are very mild, because living things live in, you don't reflux bacteria, for example. So the enzyme that does this contains a diiron cofactor, which is shown on the right, but the full protein is shown here. It's actually a dimeric protein, so there are two active sites, so one here and then one right here. If you look closely at the iron centers, you can see that it has these irons that are ligated by histidine side chains as well as carboxylate residues. So you've got two carboxylate residues that are bridging and then two that are terminal, and here it's just a water molecule. And this is the reduced form of the protein. So one of the main projects that I was working on, and this is effort in my research group, I worked for Steve Lippard at MIT, who's now currently retired. But he spent about 30 decades studying this enzyme because it's extremely complicated. And so my PhD project was basically just to see if we can reproduce this cluster structurally in a small molecule, and then more importantly, can we use that to see if we can reproduce this chemistry in the lab. So kind of summarizing my 5-year PhD in a couple of slides, here is kind of the highlights of what I had done. If you've ever done any synthetic chemistry, especially working with inorganic complexes, you know that if you take just iron salts and you add these ligands into the reaction flask and stir it up, you're just going to get a mess because it's not selective, right? You'll get many different species, you might not even be soluble, and so that's not going to be very useful for making a structural mimic. So I made actually a lot of different ligand designs, but this is the one that worked the best. I decided that to kind of achieve the same structure, I wanted to first have a macrocycle so that we can kind of at least mimic the donor atoms, right? So you've got your oxygen donors, your nitrogen donors, but because of the macrocycle, it provides kind of a pre-organized platform for accessing the core structure. So I was able to synthesize this imine macrocycle in the lab. I can add in 2 equivalents of iron and salt, as well as 2 equivalents of carboxylate. And you can see here's the X-ray structure of that product. So the irons are now bound to the oxygen and nitrogen chelates that are shown here, and then you have a carboxylate that's on the bottom face of the structure that's bridging, and then a carboxylate on top that's also bridging, but in an eta-2, eta-1 kind of bridging motif. So what's really exciting about that result was that I compare the X-ray structure of the protein, so this is from the protein, the SMMOH reduced form, versus the synthetic complex that I had made. This was actually the first structural mimic of this enzyme. So you can see that the nitrogens are both bound in a cis arrangement, and then you've got these bridging motifs that also mimic the natural environment. I did my chemistry actually in absence of water, so that's why we don't have an additional water ligand, but I think if you added a small equivalence of water, you'll probably get binding with the oxygen at this position here. And I want to point out that this is only a structural mimic, meaning that I haven't been able to reproduce the chemistry. But there are some critical differences, right? Because these oxygen donors over here and over here are actually phenolates, and they're not carboxylate groups, so it's not actually exactly like the protein. In addition, the nitrogen donors here, you go back, are imines, and imines chemically, even though it has a nitrogen donor, is very different than an imidazole type nitrogen donors. And there are differences in bond distances and bond angles, right? Those are the two iron distances is 3.43 angstroms. In the synthetic complex is 3.61, so it's a little bit further away. However, I think this is still a important synthetic achievement, because at least we can mimic the structure, and perhaps we can tune and modify this complex to achieve function. So this is kind of where I stopped in my PhD. I didn't have enough time to continue and to modify the system. But so far, us as well as other research groups have not been able to reproduce this reaction yet. So this is still one of the this is the holy grail for alkane oxidation, to try to mimic the function of this enzyme. So that's all I had to say. This is the end of this bioinorganic lecture. So thank you very much for listening, and I hope you found some of this interesting.